Gordon Berman, Associate Dean of Research. It gives me great pleasure to welcome you to this year's Chappel Memorial Lecture. I'll be giving you a little bit of a background to the history of the lecture and also an introduction to Dr.Tyrrell, who's our speaker today. The Chappel Memorial Lecture was established in 1988 through an endowment by Dr. Clifford Chappel in memory of his father, Herbert James Chappel. Since that time, internationally renowned researchers have been invited to the University of Guelph to summarize their current work and discuss the development and progression of their program over the years. The lecture and informal meetings are meant to act as a source of inspiration for those engaged in or considering graduate studies in biomedical research. I wanted to deviate a little bit from my normal introduction, and in the spirit of our 150th anniversary, I'd like to mention a few aspects of Dr. Chappel's connection to the OVC, to biomedical research, and acknowledge his entrepreneurial spirit. Dr. Chappel graduated from the OVC along with his brother Reginald in 1950. Dr. Chappel has had a distinguished career in medical research and business. He began his career as the Director of Biological Research at Ayers Laboratories in Montreal and was the founder of BioResearch Laboratories, a contract research organization in Montreal. BioResearch was acquired by Charles River Laboratories International in 2004. In 1985, Dr. Chappel co-founded CanTox Incorporated, which is a science-based consulting firm specializing in expert advice on toxicology issues. CanTox was acquired by Intertech in 2010. Among his scientific accomplishments is the discovery of the BB rat, the first animal model for juvenile diabetes. These rats are now raised in colonies worldwide. Dr. Chappel made a significant contribution to the medical community through his discovery of acepromazine, a widely used tranquilizer in veterinary medicine. For these achievements, Dr. Chappel was honored in 2003 with the OVC Distinguished Alumnus Award and the U of G Alumnus of Honor. Reflecting on his career choice, Dr. Chappel said, "I grew up within a stone throw of OVC. On my way home from school, along with my chums, we would stop by the vet school. The large animal surgery at that time had a large ground floor window where little boys could watch surgery. That's what convinced me of what I wanted to do." Dr. Tyrrell, it gives me great pleasure to welcome you. Thank you, Dr. Tyrrell, for accepting the invitation to give this year's Chappel Memorial Lecture. Dr. Tyrrell asked me to keep his introduction short, which has been quite a challenge given all his accomplishments. Dr. Tyrrell is the founding director of the Nikoshing Institute of Virology at the University of Alberta, where he holds a CIHR GlaxoSmithKline Chair in Virology. He is a professor of medicine and biochemistry at that university, as well as being the chair of the Department of Medical Microbiology and Infectious Diseases and the dean of the Faculty of Medicine and Dentistry. True to his agricultural roots, Dr. Tyrrell originally intended to be a veterinarian, he told me last night, but because OVC was the only veterinary college in Canada at the time, that would mean leaving Alberta, so he decided on another career choice and studied medicine at the University of Alberta, where he received his MD degree in 1968, went on to do a doctoral degree in pharmacology with Jerry Marks at Queen's University, and then did fellowships in internal medicine and infectious diseases at the University of Alberta, and a postdoctoral 2 years at the Karolinska Institute in Sweden. Dr. Tyrrell has received a number of awards and honors, including the Alberta Order of Excellence, he's an officer of the Order of Canada, member of the Royal Society of Canada, Distinguished University Professor awardee at the University of Alberta, and he was inducted into the Canadian Medical Hall of Fame in 2011, last year at this time. He's held numerous advisory positions in healthcare at the provincial and national level. Currently, he's on the Health Canada Science Advisory Board and is the chair of the Gairdner Foundation Board. Since 1986, his research is focused on viral hepatitis. He's utilized and developed unique animal models of viral hepatitis and has developed his intellectual property into practical applications. For example, his work, supported by CIHR Glaxo Canada, resulted in the licensing of Lamivudine, which is the first oral antiviral drug used to treat chronic hepatitis B infection. Today, Lamivudine is licensed in over 200 countries for the treatment of that disease. Dr. Tyrrell holds 24 patents and has been involved in 10 biotechnology companies as either scientific officer, member of his board of directors, or on the executive. 6 of these companies are spin-offs from the University of Alberta. I will mention 2 of the companies based in Edmonton. He's the CEO and Chief Scientific Officer of Virex Incorporated, which develops therapeutic vaccines for hepatitis B and hepatitis. He's also co-founder and vice president of KMT Hepatech Incorporated, which uses a hepatitis C mouse model, which he'll talk about today, to evaluate small molecules and vaccines. He has held many grants, research grants, over the years. Currently holds 3 CIHR grants, 1 NIH grant, and an Alberta Heritage Foundation for Medical Research grant, and a $2.5 million Li Ka-shing Foundation Award for the Institute of Virology. In fact, his latest CIHR grant, you might not know this, was approved when I was on the Pharmaceutical Sciences Review Panel. We reviewed your grant, and apparently it benefited us both because it gave me the idea of inviting you here today, and thank you very much for graciously accepting that invitation. So it's readily apparent that Dr. Tyrrell continues to have vision and foresight in leading research on viral hepatitis with a commitment to putting his ideas into action. As an indication of his visionary spirit, last night at dinner he predicted that the Edmonton Oilers will be the next Canadian hockey team to win the Stanley Cup. Maybe next year.
I remember when I was a kid, we used to go to church. We always went to the back of the room. And we went to a hockey game. We always came in very early and got to the front. This is obviously a very religious crowd. Let me say that I have really, and sincerely mean it, that I have enjoyed my visit to Guelph. It's short, but you know, when you visit a place, you can quickly tell whether people are happy where they work and they're passionate about their work. And I got to say that every place I went today, I felt passion and I felt people happy and enjoying what they do. And that's a very nice sign, and I just want to say the friendly people, it started with the hotel. The Delta Hotel staff is the friendliest staff I've ever seen in a hotel, and it continued here today. Thank you very much to Dr. Chappel and his wife who endowed this lectureship. And when I looked at the people who have been here, I feel very honored to be here and to be giving this lecture today. I'm briefly going to tell you that my talk is a little bit long, but I will get through it, and we'll try and do it in an hour. Let me just say that I'm going to have a brief introduction about viral hepatitis. And then from there, I'm going to move into work we did on hepatitis B, and then I'll move into some work we've done with the mouse model, because I've had a lot of questions about it, and so I did some modification last night after dinner. I also want to thank Guelph for David Evans. Some of you may know David Evans or remember him. But David is, when I was the dean, I recruited David as the chair of MedMicro. And David has been a remarkably good chair and a wonderful colleague that I continue to work with. He is the associate director of the Li Ka-shing Institute of Virology. Persistent viral infections really, to me, fall under 3 major categories. In Canada, we have about 45,000 people with HIV. We have between 240 to 300,000 with hepatitis C, and probably well over 300,000 with hepatitis B. And in the world, there's about 40 million with HIV/AIDS, about 130 to 170 million with hepatitis C, and 350 to 400 million with hepatitis B. And these are major diseases in the world, these persistent viral infections. And another way to look at this is you can ask the question, are you 1 in 12? Because 1 in 12 people in the world are a carrier of either hepatitis B or hepatitis C. So it's a very, very common disease worldwide. My life changed and my research changed, and I was preparing a lecture, but this is a picture of Brock Bloomberg. I don't know if I can get the lights down a little bit in the front, do you know? Sorry, I'll let you do that a little bit because there'll be some other slides that'll come up. But just take the spotlights off. Yeah, that helps. Brock Bloomberg discovered hepatitis B, and show the picture because he passed away in 2011. He was a Nobel laureate in 1976 when I was a postdoc in Stockholm, and actually got to attend that Nobel ceremony and meet him. It was quite a nice opportunity. Hep B is a virus that is present in the blood, often very large amounts. 10 to a billion copies is not uncommon in patients that carry hepatitis B, and it has the three different forms you see here. Bloomberg discovered the surface antigen of hepatitis B that can be in these sausage-shaped forms or these round small particles, 22 nanometers. But the virus particle is a Dane particle, discovered a year later by Dr. Dane working with Bloomberg. Carries the name of the Dane particle, and it has an envelope and core protein around the DNA of the virus. It's a relatively simple virus. Around the world, when I say there's 400 million people carriers, most of them are in Southeast Asia, China, very common in China, Sub-Saharan Africa, and parts in Northwest Territories and the northern parts of Canada and Alaska. And the virus can be transmitted a number of ways, but the way I wanted to emphasize most today, because where the carrier rates are so heavy, is transmitted from a mother, and the mothers, about 50% of them have the infectious virus in their blood when they're a carrier, and they'll transmit that virus 100% to an infant born to the mother. And that, if a baby's infected at birth, they can't get rid of the virus very easy. 90 to 90% will become chronic carriers. If you're infected as an adult, it's 3 to 5%. But as an infant, it's 90 to 95% end up as chronic carriers. And if you're a male and you're carrying hepatitis B infected at birth, Bloomberg studies in Taiwan showed about 50% of those males would eventually die from hepatitis, either cirrhosis or liver cancer. In females, the disease is less aggressive. They go on, they have their children, and then about 15% of women later in life will develop the complications of hepatitis B. So the virus discovered by Bloomberg carriers, it is the most common cause of fatal cancer in the world. Hepatitis B causes the hepatomas, and the hepatomas are hepatocellular carcinomas, are often diagnosed late, so there's a low cure rate in most hepatomas. We do a better job of screening carriers now and picking up the hepatomas earlier, but I came in last night, it doesn't seem long since we had dinner. Between last night and tonight, there'll be 3,000 people in the world die as a result of hepatitis B or hepatitis C. So these viruses are still, we don't see them as much in Canada, but worldwide, they're big problems. We have a very effective vaccine for hepatitis B, and the real issue is not the vaccine, but how the health policies use the vaccine. In Canada, for a long time, we only vaccinated firemen, policemen, doctors, nurses, lab technicians. But in fact, the incidence of hepatitis B continued to increase in Canada, even though we had a good vaccine. And you know, when we introduce any other vaccine, you see the virus and the disease go down. And we had to change our policy to vaccinate young people to catch up, usually grade 5 to 7. I hear it's 7 in Ontario, 5 in Alberta. And we are vaccinating anybody who's exposed to the virus early in life. If there's a carrier in the house or the mother's a carrier, the baby gets vaccinated at birth. Taiwan is the first country in the world that vaccinated every baby at birth, started in 1986, and they had a significant incidence of hepatocellular carcinoma in children. And since they started the vaccine program in infants at birth, the incidence of carrier rates in Taiwan in children have gone dramatically down, and hepatocellular carcinoma in children has virtually disappeared. This is truly a vaccine that prevents cancer, and it should be used more than we use it. The end result of hepatitis B or hepatitis C carriers is often hepatocellular, a lot of fibrosis. Liver should look normal. This is a liver that we took out of a patient that had a liver transplant, and you can just see the regenerative, the scarring, and it's very difficult for blood to flow through that liver because of the cirrhosis. And many of these patients die from esophageal varices bleed, as the end event in their life. The other disease is hepatocellular carcinoma. This is a 38-year-old man from Hong Kong that was a patient of mine that had developed this hepatoma. He had it removed. He lived 8 years and then had another hepatoma and unfortunately died. His son went on and graduated in medicine and is working as a physician. My original research was on MS and multiple sclerosis and paramyxoviruses, particularly measles. And I was working on this from 1976 to about 1986, 10 years, had a CIHR or MRC grant at that time. And I was preparing to lecture to our graduate students in a new course in advanced virology. And I read the papers on the replication of hepatitis B. You know how it is when you're teaching, you have to try to learn it a little better than your students and stay one page ahead. And I was trying to do that, and I got very fascinated with this because there had been a publication just a couple years earlier by Jesse Summers and Bill Mason on the replication of hepatitis B, and it's the only DNA virus that goes through an RNA intermediate. So it goes from DNA to RNA back to DNA. And it has, the viral polymerase has reverse transcriptase, RNase H, and DNA polymerase activity, all the same activities you see in HIV. It has one other additional activity. At the carboxyl terminal end of the virus, or the N-terminal end, you have a protein priming function. And the DNA of hepatitis B is primed by a protein. DNA synthesis in us is all by RNA priming, and this is primed by a protein. So a little different. And when we were doing this, we thought maybe there's a way we can inhibit this virus with drugs. And so the virus came in. Jesse Summers' paper essentially showed the virus coming in, gets uncoated, the core goes to the nucleus, discharges the DNA, and it's partially double-stranded DNA here, and it gets this protein that's bound to the N-terminal end of the DNA gets cleaved off, and the seed gets converted to covalently closed DNA. And this acts as the template for transcription of messenger RNA and the template for transcription of a pregenomic RNA. The messengers go out to make proteins. They form what we call replicating cores, and you have a polymerase gene plus the RNA gets inside the nucleus or gets inside these replicating cores. And you can prepare the replicating cores from liver of ducks that are infected or animals that are infected and study the replication. And Jesse Summers used this to show and work out the replication model for hepatitis B. And it comes back, this is an RNA template. That's used to make the DNA on the first strand, and then the DNA serves as the template for the second strand, and you get the double-stranded back DNA and virus. So this was 1986. There was no cell culture system, a traditional cell culture system you could use. No animal model other than a chimp. But there was, had been discovered early in the 80s, the Peking duck and the woodchucks that carry the virus. I have worked with the woodchucks. The woodchucks are a wonderful model because they have actually 4 genes. They have the surface, the core, the polymerase, and an X protein. The X protein is a transactivating protein that does a lot of things in the cells, but animal viruses for Hep B cause cancer, and the avian species do not, and the avian species don't have an X gene. So I just wondered, could we use the duck system that had been used to work out the replication to try to find if we could use that as a surrogate to look for antiviral compounds for Hep B? And one of the other things you learn very early in your career is if you want to be successful, try and associate yourself with very good people and successful people. And I had lunch after preparing and giving the lectures. It was bothering me, and I went over to meet a nucleoside chemist at the University of Alberta by the name of Morris Robbins. Morris was an outstanding nucleoside chemist. He was the first to synthesize the dideoxy nucleosides and the purines. People couldn't make, they could make the pyrimidines, but they had trouble chemically making the purines. And he was the first to synthesize with his uncle the DDA and DDG. And Sanger, in coming up with the enzymatic sequencing, needed these two and got them from Morris at the University of Alberta. He provided the dideoxys for Sanger's ideas. I always thought he deserved more recognition for this as well. This is a picture of Morris. Then we had lunch together in the faculty club, drew out that replication cycle you saw, and I said, you know, Morris, some of your compounds I think would inhibit this virus. Can we work together on this? And he was very enthusiastic. I got to say, it was we worked together for 13 years, and never a cross word, just a wonderful, wonderful colleague. He was a chemist, I was more a virologist, but it seemed to be that when you got together, and you know how it is with some colleagues, you get together, there is a magic that seems to happen, and within minutes we were throwing ideas back and forth. It was just a great collaboration. This is my other collaborator, the Peking duck. The Peking duck is the one that Jesse Summers had used for that replication, and it's a great animal, inexpensive. When it bites you, it doesn't really hurt, and we use it a lot. Now, I got to tell you, in the beginning, I had no money to do this, but I had my parents. I lived on a farm, raised on a farm west of the city, and I was getting some ducks in from California. What happened is I was getting them from Sparfus, Pennsylvania, originally, and they sent them in, and everything was okay. And then I got a phone call one morning at breakfast that the barn had burnt down the night before, and it was an older couple, and they said, we've lost our ducks, and we don't have any more ducks we can send you. So I was desperate because it was wintertime, and we were excited. We were getting some good results, and I found out that a place in Gonzales, California could send the ducks. So I said, how are we going to get them? And he said, well, I'll just send them through the mail. So he sent them through the mail. I got a dozen, they were fine. He sent another couple dozen in a box, sent to me, and fine. I ordered 5 dozen, and it was a warm day, and I got a phone call from the post office to come down there and look after this box because it was wet and sticky, and it had my name on it. And I went down to the post office, and I got to open it up, and out of the 60 eggs, there were 11 that weren't broken. So I cleaned up the mess. It was at lunchtime. People were sitting in their lunchroom. I cleaned up the mess and took my 11 eggs and walked out. Went back to my lab. And about an hour later, I got a phone call from Agriculture Canada. And they said, what in hell do you think you're doing? And I said, why? And they said, you're not allowed to bring those in. And I said, I thought it was all fine. No, you got to return them immediately. So I was desperate at this point, and I got a dozen eggs from Safeways and returned 11 to them. And we managed to carry on our work. And this is at my parents' farm. I took the ducks out there, set up a little colony at my parents' farm, and they raised them for 2 years later, and I'd go out on weekends and pick up the eggs and incubate the eggs and then use the ducklings for the work. Now what we did is if you get ducklings early infection, you could see the virus increasing as we had the cells in culture. And the darker the dots, the more virus. And this is one of the early experiments we did with some of Morris's compounds. And we had dideoxythymidine, day 20, no effect. Dideoxycytidine at 10 micrograms per mil, there's some effect, but it was a bit toxic. At 1 microgram, nothing. ERA-A was another compound we've been trying in hepatitis B, and it had some activity, but not great, but it had some. But what surprised us and was delighted to see that DDG, particularly, was a very potent antiviral agent, more than 10,000-fold lower in this cell culture system with DDG. So with that, we wondered why DDG was very potent and DDC and DDT were not. DDA is fairly potent. I won't go into the details. It gets metabolized to DDI and isn't very good orally. But DDG, we had a prodrug made by Morris called 2,6-diaminopurine. And the 2,6-diaminopurine was quickly deaminated into DDG. And the DDG then, the first nucleotide binding to this primer protein is always deoxyguanosine. And if you put a dideoxyguanosine on there, it can't continue the chain synthesis. But if you put DDC out here, it might get proofread out. So I thought there was proofreading for the binding between nucleoside-nucleoside bonds. So DDC was getting proofread out, and DDG was binding to that primer protein and wouldn't get proofread out. And I can tell you that we did the experiments to show that DDG binding to this protein cannot be cleaved out by the normal mechanisms. We then went into animals, and we took 12 animals infected with hepatitis B, ducks, and these are serum samples and just a dot plot for the amount of virus. These were all carriers, and we simply treated them for 3 weeks with either placebo, DDA, or DDG. And you can see after 1 week, almost all gone, 2 weeks, 3 weeks. And this is the first time we'd really seen that there were compounds that inhibited hepatitis B as dramatic as this. And so we thought that it was great. We sent it off to Nature, quickly got a reply that it wasn't going out to referees because it's not of sufficient reader interest, even though there's 400 million people, and there were 2 million people at AIDS at this time. Everything about AIDS got published. Hepatitis B, not sufficient reader interest. Anyways, won't go into that. We later published it, quickly published it then after it had been out in Antimicrobial Agents and Chemotherapy. Then we looked at, I won't go into all the details, trying to start a company, didn't be able to start a company at that time. Then we looked for companies to partner with. We looked at Merck, we looked at Lilly, we looked at Wellcome, and we looked at Glaxo. And Glaxo was the one that we ended up partnering with. And this is a few years later when they opened their new facility close to here in Mississauga. And working with Glaxo, we'd gone to a Gordon conference and seen some work with Lamivudine, 3TC, and our work was presented. And we asked Glaxo if we could get some of the Lamivudine or 3TC to try in our model because of the nature of replication of hepatitis B being similar to HIV. And we screened a lot of compounds for Glaxo, but in this lot, these were done in duplicate. And you can see there are these compounds, 18, 20, and 22, that seem to be good inhibitors of the virus on the screening process, and they all turned out to be different lots of Lamivudine. Now, I just want to say that Lamivudine was synthesized first by Bernard Boileau at McGill University, and Biochem Pharma was established. And Lilly Glaxo had licensed to Lamivudine from Biochem Pharma. So we did some work on plus and minus enantiomers. And the plus enantiomer was quite toxic to cells and did not inhibit the virus. The minus enantiomer was a very effective antiviral agent, but didn't cause toxicity. So purely by luck, but that's how these things worked out. So it was synthesized, Biochem Pharma had it, they licensed it to Glaxo, and Glaxo we approached to get this to try it. We were interested in the mechanism because this is a pyrimidine derivative, not a purine, so that must have a different mechanism. And when we did some work, I won't go into a lot of the detail, but just to show you that in an isolated system, it inhibited the same as DDC. And in a purified system, not in these replicating cores, but in an enzyme system, you could look at it and you could see that this was being incorporated wherever there's a DDC or a cytidine in the DNA, 3TC was incorporated. And we published that on the mechanism. But I just want to say there's still a little mystery because why did this get incorporated and not get proofread out? And we still had no way of knowing for sure if there was proofreading with hepatitis B because it didn't have the typical domains of an endonuclease or a polymerase that had proofreading activity. So hepatitis B, when it's synthesized inside these cores, is actually quite a small volume, very tiny volume. And the cores have a number of lysines on them, and it keeps minus charged molecules inside it. And I just want to say that DNA synthesis, we always think of DNA plus a nucleotide, DNA polymerase, it gives you a lengthener by a nucleotide plus pyrophosphate. But the pyrophosphate, if you build up the concentration, can actually reverse this reaction. You don't think of this being reversed very often, but it does reverse. And we did some studies. I had a great student who walked into the lab, wanted to work with us, and took this project on. And he showed that if you terminated the DNA, you put in radiolabeled nucleotides, and then terminated it with DDC, 3TC, or terminated with any of these, and then tried to proofread, the 3TC got incorporated but wasn't getting proofread out. And we published a paper on phosphorolysis and the fact that 3-TC, or lamivudine, gets incorporated but is resistant to phosphorolysis, whereas the others were proofread out. So in effect, you had an ability to selectively inhibit the virus with lamivudine. And it gets incorporated by the viral polymerase, but not by the human polymerases. I would point out that we went back then to see if this was going to happen, and we had this all in duck. We wanted to look in human. We looked at lamivudine in the ducks, and this is hours on the side here, and 3 ducks treated with lamivudine and 3 with placebo, and just taking serum samples every 8 hours for 72 hours, and then looking at the dot plots, you could see how rapidly the virus disappeared. I really felt at this time we were going to cure chronic hepatitis B. And the excitement was fun. We went to human virus in chimps. Robert Purcell at the NIH had done a bunch of studies on chimps and developing the vaccine, and he used some placebos and then exposed them to these chimps to the wild, to the virus. And of course, some of the chimps were infected, and a few of the chimps became chronic carriers. This was done in New Iberia, Louisiana, and they had 68 chimps. So I screened the chimps that had been in those studies, and we found 3 chimps that were carriers of hepatitis B. And we got infants from those chimps. They had young infants, and they were also carriers. So it had transmitted vertically, just like it does in humans, to these young chimps. And this young chimp, you can see, they were well treated. I mean, these chimps had Fisher-Price toys all over. You came in the room, they'd hop back into their pen. That was their protected area. But they would have diapers. They had nurses looking after them. And they drank Tang twice a day. Come in with a little glass of Tang, and they'd take it in their hand, and they'd drink it. And we spiked their Tang with lamivudine, see what would happen to their virus. And I was down there for a while, and we got samples of blood from them, and 0.1 milligram per kilo of lamivudine, 0.3. We just kept tripling increasing the dose, and then came back and analyzed what had happened to the Hep B in these chimps. This chimp, which had 350 picograms, which is well over 1 billion copies of virus per mil, even with 0.1 milligram per kilo, and then at day 14, we increased it to 0.3, and you can see we dropped it down to undetectable virus at that time. So we knew it worked in humans, and we quickly got permission, Glaxo got permission to go into humans, and we treated humans with placebo, 5 milligrams a day, 20, 100, 300, or 600. And every human in the study for 28 days that got 100 milligrams or more. 100 milligrams is 1/3 of a regular aspirin size. So it's about that much we were giving them once a day. You can see that the virus had dropped by 99.9%. This was greater than a 3-log drop with this. These studies were done very early in about 1990, and they did some more studies, and lamivudine 100 milligrams became the dose that we standardly use in hepatitis B. Also put this paper into New England Journal, think it was exciting, and got the reviews back that we should have done biopsies on the livers before and after, before they would consider it. And there was another individual that was then working with lamivudine from Harvard, and a few months after our paper was turned down by New England Journal of Medicine, it was published without biopsies in the New England Journal of Medicine. So I had presented it at the liver meetings in Chicago before that, so I keep saying this is the first publication on the human use of lamivudine in hepatitis B. It's 1993, and it was a phase 2 study showing you those results that we had. We later published other papers. Let me just say that having a drug that inhibited the virus, could it reverse the disease? And we started to see patients that were on therapy seemed to be getting better, and biopsies were done. And this is a biopsy, this is a picture taken from Ian Wanless at the University of Toronto. It was a friend of mine that had sent me this picture and said, this is just being published, but you can see what your drug is doing. And this was a patient that was on lamivudine for 2.5 years with no resistance. And this is a cirrhotic liver in the biopsy of pre-treatment. And these 2.5 years later are two biopsies that he called normal. It really changed the liver a lot. And we see this because nowadays you put patients on an antiviral agent, getting ready to transplant them if they get end-stage liver disease, and we see a number of those patients actually improve and come off the transplant list because the antiviral agents can reverse the disease. There's another study that was done in 1999 just showing that the patients that were on placebo had a lower survival rate than those on treatment, and when you looked at hepatocellular carcinoma, on treatment in yellow and in blue was the control group. And there were several studies done in patients with advanced liver disease and Hep B that the studies were cut short because it was felt it was unethical not to treat these patients, and we had good statistics for supporting treatment. Just to show you that in chronic carriers, the viral load is very important, and the higher the amount of hepatitis B, the higher the incidence of hepatocellular carcinoma or the progression to cirrhosis. So the objective of all this therapy with hepatitis B is let's get the viral load down as low as possible, let's get the inflammation out of the liver, and the liver enzymes normal as soon as possible, and that's the basis of therapy today. This is Lamivudine, Heptavir as it's known in Canada, that was licensed in 1998 and since then is licensed in many other countries. When we were doing this, Carl Fisher in my lab had taken the polymerase gene and the primer protein's N-terminal end and the carboxyl is RNase H activity, middle is the DNA polymerase activity, and there's nucleotide binding sites in here. And compared to HIV and all of these viruses, there's a domain in the nucleotide binding site, the YMDD motif. And in hepatitis B, we simply mutated the methionine to a valine or an isoleucine and looked at the binding and the activity. The wild-type virus, untreated, treated with DDC, no activity, treated with lamivudine, completely inhibited. The mutated virus, YMDD to the YVDD, and that binding site didn't replicate as well. And that's not uncommon when you make these mutations, that the viral fit isn't as good and they don't replicate quite as well. But no activity of DDC and certainly no activity of 3TC. And we were concerned at this point it was going to be fairly easy to get resistance to lamivudine, and wrote this article, and the last sentence said, this report of a genetically engineered lamivudine-resistant hepatoviruses serves as a cautionary note of what could occur when lamivudine is used prolonged treatment of chronic hepatitis B. And shortly after we published this paper, and lamivudine was in clinical trials, the first lamivudine-resistant viruses were reported. And it's not uncommon to have lamivudine resistance. So it was used for a number of years, but there are better drugs. Lamivudine was introduced in 1998. Adepavir in 2004, in 2005, entecavir, in 2008, tenofovir. So there is a family of drugs now for hepatitis B. Lamivudine is safe, inexpensive, effective until you get resistance. Fortunately, tenofovir and entecavir will still treat the resistant virus. These are the two drugs that are the best now because they have less resistance. But you'll note they're both purine analogs, and they both act by that blocking the primer protein. Just looking at hepatitis B with lamivudine, after 4 years, about 60% of the patients that are on therapy all the time, and it really depends how high the viral load was at the beginning. High viral loads, more likely to get resistance. Low viral loads, or if you can suppress the virus completely, then no virus, you don't get resistance very quickly. But patients that are on lamivudine develop resistance. Adepavir, they develop resistance. This is another one that is developing resistance. But entecavir, the resistance is very low. And tenofovir, there's virtually no resistance. So they have become the drugs of choice in treating hepatitis B because of the resistance problem. So lamivudine was the first oral antiviral agent. It's actually licensed over 200 countries, I should have put in there, but cumulative sales now are over 5 billion. It's the most commonly prescribed drug in China. problem was antiviral resistance, and the newer antivirals have much less resistance, as I said. This was the team that I worked with in developing antiviral therapy for hepatitis B. Morris Robbins, who I've mentioned has been key, a liver pathologist, Lawrence Jewell. This young lady was from China, Weijing Liu. She's now in a biotech company in California and doing very well. She stayed in Canada and in the United States. And you can tell this was many years ago, because look at that guy I didn't know. But it was a good team that we had together at that time. I won't go through all the names in here, but I would say that Weisheng Liu and particularly Bonita Lee, these two people were tremendously good students that made contributions to how this drug worked. And we were supported by CHR, Alberta Heritage, University Hospital, and Glaxo was a very good support. I would say that working with industry and Glaxo over those 13 years, I've never once had Glaxo come in and say, you must do this or you should do that. They left me working on my own. We had the same objective, was to try to get antivirals that worked against hepatitis B. So I'm going to touch a bit on hepatitis C. Hepatitis C, we had the animal model. We could see how valuable it was in trying to do work with hepatitis B and getting these antivirals. And we wanted to do something similar in hepatitis C. There was no cell culture system in 1998, no animal model for hepatitis C except a chimp. And there are major problems working with chimp from ethical points of view, etc. So it was hard. And I want to just acknowledge at the very beginning two people, Norm Kniedemann, who was a liver surgeon and transplant surgeon at the University of Alberta, and he had a graduate student by the name of David Mercer, who was also a surgeon who did a PhD on this project, and I was on the supervisory committee. And one of the supervisory committees, he showed this slide, and he said, he was putting in human hepatocytes, and this is human albumin in the mice, and these are skid beige mice, so they don't reject human cells. They have no ability to reject them. He put them in the lower pole of the spleen, 1 million cells, and within an hour, most of those cells have moved into the liver and repopulate the liver. But you can see that with time, they just died out. They were not going to stay, and this is a marker of human albumin in the Western blot. So I suggested to him at that point, I said, when we farm, if we don't spray the weeds, we don't get much of a crop. And he said, what do you mean? I said, well, we got to get rid of the mouse hepatocytes some way, so that it gives the human hepatocytes that you're putting in a chance to expand and grow. And we were thinking of different ideas. If you take an overdose of Tylenol, you can rescue these people with N-methylcysteine if you get them early enough. So what I had thought about originally was, let's poison the mice with N-methylcysteine, and then go in and give them, poison them with Tylenol, acetaminophen, rescue them with N-methylcysteine 12 hours later, give them a bunch of human hepatocytes and see if that would work. And we started to do that, and then Carl Fisher, again in my lab, had gone to a meeting, and he'd heard a presentation by, I'm sure somebody in this room, a few people in this room will know Brinster, was a veterinarian who recently won the Gairdner Award for his work on transgenic animals and the animals he made. And Dr. Brinster's had these papers in Cell about neonatal bleeding with a transgenic mouse model that knocked out liver cells. And his transgenic mouse had an albumin promoter and urokinase behind it, 4 copies. And overexpression of urokinase in these mice livers causes the mouse to lose its liver function, lose a lot of liver function. So instead of using the acetaminophen and trying to rescue, we put this transgene into our mice, and we crossed them, did some crossing to the skid beige, and ended up with mice that were carrying the transgene as well as the skid beige. And this is pictures of the cell of the livers from animals. And if the animal's homozygous for that transgene overexpresses urokinase, the liver turns out to be very pale, sick liver. This is a normal mouse liver, and these are the hemizygous. And some of the hemizygous cells will lose the transgene and give you normal cells, so you get the spotted liver and the hemizygous animals. And these mouse cells that lose the transgene take over and start taking over the liver and replacing it all. What we were doing was we were taking samples of liver tissue from patients that were undergoing liver surgery, resections, and putting in, digesting them and getting cells that we could infuse in the lower pole of the liver. And lower pole of the spleen, pardon me. And this is an older mouse, but we usually use the younger mice. Just make a small incision, get the spleen out, infuse 1 million cells very gently into the spleen, put a small clip on it, and drop it back into the abdomen and close them. And they move into the liver, and this is human cells growing in the liver. And all the red areas are in fact human cells growing in a mouse liver that are like this. And I will just show you an animal that we did multiple laparotomies on to show how the liver changes over time. And there's an animal that was transplanted, and 6 weeks later you see these human cells, but there's spots of human cells all over. And at 12 weeks, that same animal, these have grown and these others are growing. And by 17 weeks, most of the liver is humanized. So we ended up producing little mice with human livers. And then we tried to infect them with hepatitis C. Remember how I showed you the albumin disappeared? Now when we put in human cells into these, the human albumin signal became stronger. And these are a bunch of animals that carried the transgene and those without, and they're all transplanted with human cells the same. So the transgene made a huge difference on how successful the transplants were. And we infected 22 animals, thinking we were going to get 22 animals with Hep C. I've skipped over some things here, but what we found was one animal. I keep thinking back, if we'd have never found that one animal, we'd have given up on this. It was not going to work. But we found one animal that had hepatitis C, and the amount of hepatitis C it had was very similar to what you see in a human carrier. It was about 10 to the 6 copies per mil. So it gave us great encouragement. I mean, if you hear a talking dog, you know that a dog can talk, right? But you have to hear one. We went back and analyzed that mouse. Why was it different to all others? Why did it take it? And I can tell you that Bill Addison in my lab did real careful genetic studies. And this is the endogenous transgene, or the endogenous urokinase gene. And this is the transgene we put in. And in a hemizygous form, it's here. In a homozygous form, it's here. And in all the mice that we looked at, those 22 mice, 21 of them were hemizygous and 1 was homozygous. And only the homozygous animal took the transplants, or took the infection. They all took some of the transplants. And I'll just show you that this is human alpha-1 antitrypsin, and the homozygous animals have much higher levels and better engraftment than the hemizygous animals here. So we went back and redid our animals to make sure they were all homozygous. And then we infected these animals. These are 4 animals we infected with hepatitis C, and these are weeks out. People had put some human biopsies with hepatitis C under capsules of mice and trying to get them to grow the virus so you could do an assay. It would come and disappear. These animals got virus. It's up there in 2 weeks. When you infect them, it goes up rapidly, and it stays at nice levels throughout. So you can do antiviral studies in these animals and see what your antivirals will do to the level of virus very easily. And this just shows you the engraftment. We did a lot of animals, but most of our animals infected with hepatitis C, we would get pretty good levels of virus. These are lower, 10 to the 4, 10 to the 5, but 10 to the 6. So we had an animal model that worked, and we could infect with hepatitis C. Just showing you that these the human cells engraft into the mouse very nicely, and they hook up with the biosystem, and there's no problem with any of that. And we submitted this to Nature Medicine, and it did get published, but they wanted us to do transmission studies and some other studies with negative strand virus to make sure it was really replicating. And this just shows a 37,000 increase as we passed the virus mouse to mouse from what we started with inoculum. So we knew that it was growing in the mouse, and it was accepted and published in Nature Medicine in 2001. We did set up a small company, and that company is called KMT Hepatech, and we provide services to companies that want to test their compounds in the mouse model. We infect them with hepatitis C. We also do it with hepatitis B, and this animal also supports malaria, falciparum malaria. There's no animal model for that. And it can grow, you can grow malaria in this animal model. We actually give them interperitoneally some human erythrocytes, with the transplanted livers, and they end up getting the full cycle of malaria infection. Just want to say that we used it, we had to ask some questions, infection, prove it. We had to know how much is human, and we do a succinyl-CoA PCR. The mouse succinyl-CoA is different to the human in length, in the intron. So we can tell mouse from human and set up standard curves with these PCRs so we can tell you how much is, this mouse has got 85% human, 84% human, 72%, and this is the mouse and this is the human transgene. The darker the transgene for the human compared to mouse, we could work out approximate percentage of engraftment. It gives you a unique opportunity to study different viruses in exactly the same cells. You think about how you would do this in humans or in chimps, it would take a lot of work. But we can set up mice, a lot of mice with exactly the same cells and infect them with different hepatitis C viruses to see how they vary in their induction of innate immune responses or whatever you would like to study. You can put different hepatitis C into exactly the same, or we can put an identical virus into mice produced with different donors. So you can see how the host factors affect it versus the mouse factors. And it's a clean study to study innate immune responses because you don't have the adaptive immune response to see. Just looking at the response to infection, in minutes you get hours, you get an innate immune response, hours to days in natural killer cells, and later the adaptive immune response. And we have done a fair amount with Michael Gale at the University of Washington on the innate immune responses. And this just shows you with Sendai virus, the phosphorylation and RIG-I. And in this case, we're looking at the IRF3. And IRF3, in the presence of, becomes phosphorylated in presence of infection, forms a dimer and moves into the nucleus. Here's the uninfected, and here's the infected cells showing you the RIG-I, not the RIG-I, but the IR3 moving into the nucleus with infection. And this just shows you the, this is an innate immune response in our mice. And this just shows the interferon response genes in response to hepatitis C. And we could study that system as well in this. This is just looking at patients differ in their response to hepatitis C. And these are transplanted patients and biopsies showing either the upregulation of genes or not. And there's big differences in the innate immune response in patients. And in fact, in our mice, these are mice that are infected with the same strain of virus, but different mice. And you can see that some of them have very little upregulation and others have marked upregulation. So there's huge host factors involved in this, how you respond to them as well. And we've published this work as well. And this just shows you, again, IRF3 moving into the nucleus. And in our mouse model, where we get a good IRF3 response, as we've seen in these two IRF3, here, the phosphorylated IRF3 moving in, both of those mice cleared the virus with a strong innate immune response. And the others that didn't get it did not clear it. And another antiviral response is interferon-induced transmembrane protein 1. And I just show you how nicely the mouse model works in this. Is we've stained these for the transmembrane protein. It's there. Tight junctions stained for a specific protein is there. And superimposed, you can see that this is exactly into the tight junctions after infection and is involved in blocking hepatitis C infection in these cells. We've done work with pathogenesis just to show you that the virus can induce apoptosis. This is just looking at apoptotic cells, infected mouse, uninfected mouse. And when we look at, I'll go right here to NF-kappa B in the infected cells, the NF-kappa B is knocked down and exactly superimposed with the same cells where the NF-kappa B, wherever the hep C is infected. So we can study that type of response as well. And it does have fibrosis. Surprisingly, 8 to 10 weeks out, this is uninfected and infected, and you can see staining For different aspects of staining for fibrosis, it'll show the fibrosis, and it induces oxidative stress and apoptosis in those. This is an example of the first oral antiviral agent that was produced by BI in Montreal, Boehringer Ingelheim. This was a very promising drug, 2061, and after just 4 days of therapy, undetectable in 3 mice that we used just to show you how this is useful in showing, predicting the responses in humans. We have published some papers on how well this mouse predicts the outcome in humans as well, so it's very useful from that point of view. Just interferon response, this almost completely mimicked the interferon response in a patient, and the mouse was very close. So the mouse model, I won't go through that summary, but it's this is our first mouse that got hepatitis C. First mouse in the world to have hepatitis C, and certainly we thought we should take a picture of it. I want to just say a lot of people involved in this work. Norm Kniedemann, David Mercer, lots of people in my lab, Bill Addison, Carl Fisher, and continues to be a number of people. And we have a great collaboration with Michael Gale and Michael Cates at the University of Washington, doing a lot of the innate immune responses. And we had some trouble with the mouse that I didn't go into today. We lost some of that beige gene, and we had to do some work to get the beige gene back in. We now have very efficient production of the mice. Our efficiency dropped down to about 10% for about 2 years, and I think it hurt the reputation of the mouse because we couldn't produce enough in collaborations. We now have it up over 80% over the last 2 years working with it and getting it working well, and we're looking forward to doing a lot more work on with hepatitis B, hepatitis C, and possibly with malaria using this mouse model. I'll stop there and thank you very much.